guys, welcome back to our first video for the exam series for Biology Unit 3-4. So I'm going to work on the sample exam of 2017. We're going to work on question 1 of section B today. So I'm going to assume that you have read the question and have also attempted it. If you haven't done both, then please pause this video and at least have a look at question 1 of section B. Alright, this is on page 20. As you can see on the top. Alright, so let's go into the question. The first thing I do when I see a question that is so long is to definitely underline keywords, because you sort of get lost as you read through the information that's in the question. First up, we have cellular respiration, sucrose solution, oxygen ethanol, room temperature of 22 degrees Celsius, and these I would think are the main keywords. Of course, different people have different keywords, but that's totally okay, as long as you grasp the main concepts of this question, which is definitely cellular respiration. So once you see this word, you already should have keywords in your mind, like mitochondria, carbon dioxide, ethanol, fermentation, anaerobic, aerobic respiration. To be able to do this, you need to be really familiar with what's on your study design. So I would really advise you to go back to your study design and read through what topics are tested on your examination. After cellular respiration, what I would do personally is to label the different variables given in this question, because there are so many pieces of information that you kind of get lost towards the end of the question. And this is what I would do. I would label sucrose solution as the controlled variable. Oxygen, ethanol, the ones being measured, would be dependent variables. 22 degrees Celsius would be a controlled variable as well. So this makes me really alert and really aware of what's in the question. This is a very typical experimental type question. Experimental type questions usually give you a scenario and then ask you to explain what sort of variables are involved in this experiment, and also would also expect you to be able to explain the biological concepts that back up this experiment. Alright, on to question A. Why was it important that air could not enter the container? So again, I am underlining or highlighting my keywords here. This question addresses control measures used in experiments. So this is what I would say. Preventing oxygen inflow into the container ensures that the oxygen level measured across groups are not falsely increased by inflow of atmospheric air. There are different ways to phrase this particular answer, and there's no one way to do it. However, it's really important to identify keywords here. The keywords in this particular answer would be preventing oxygen inflow. So oxygen is definitely something you really should talk about, as well as how oxygen level actually is truly reflective of the true results. So on to question B. In order to draw valid conclusions, would you use raw data or mean provided? So again, keywords, mean, student data, percentage of oxygen, and explain your choice. Note that this is a 3-mark question, so we are expecting at least 3 points. First thing I'm going to do is I'm going to draw 3 dot points, just to make sure that I know how many points I need to put down this answer. In this particular question, you can't just explain either raw student data or the mean. You need to talk about both, because we're actually comparing which method is best. So I can start with one. I'm going to talk about how raw student data in this scenario isn't exactly the best option. I can say raw student data may not be accurate, as there may be human or measurement errors in a particular group of yeast. And then I can go in and talk about how using the mean is a good idea. On the other hand, using a mean value, okay, accounts for natural variation of rate of cellular respiration across yeast groups, because we know that not all yeasts actually operate at the same rate of cellular respiration, so it's important to pick up this point. And you can also say, and offsets errors in individual groups measured. Alright. And then finally, you can give a comparison, you can give your statement, you can talk about which one is best. So I'm going to say, using the mean is preferred over raw student data as it ensures higher experimental validity. Okay, so this is what I would say. Again, this is not the one way to talk about mean and student data. It's really up to you how you present your ideas, but it's really important to be able to compare the two groups and also give your general statement. Okay. C. Right. What factor other than the levels of oxygen in ethanol could be measured to monitor the progress of cellular respiration? So again, I'm underlining my keywords. We actually need to think about the concepts behind cellular respiration. So I'm going to write out the concepts in red, okay, to make it really easy to identify. First of all, we need to know what the inputs versus outputs for cellular respiration are. It's in yeasts, okay? So in yeasts, they carry out fermentation, which is different from aerobic respiration. Inputs include oxygen, include sucrose, which we're using in this particular experiment. Outputs would include ethanol, okay? Or carbon dioxide. So in order to measure the rate of cellular respiration, we can pretty much just put down any of those inputs or outputs we haven't already used in this experiment. For example, I can say, monitor the level of carbon dioxide production. And there you go. That's 1 mark. Simple. Question 2. Identify a factor that could be changed in the original experiment to increase the rate of cellular respiration. Again, okay, here we're thinking of what sort of variables we already have. Okay, so we have 22 degrees Celsius as our control. We have 0.1 mole of sucrose. So with this question, we think about what the limiting factors of cellular respiration are. If you would remember, it could be going back to our lessons from term 1, you should be able to remember that this includes temperature, includes substrate, which is sucrose used in this scenario, could be our other substrate, which is oxygen. So pick and choose any of these factors. I'll pick, say, sucrose concentration. Okay, could be increased. So the main point here is concentration and increased, okay? Please don't just write sucrose. It doesn't tell me anything, okay? The real important word here is concentration. You're telling me that we're changing the concentration of a substrate. So really, please put this particular word in and make your answer really specific.